Cloud. All right, so my understanding is that we are in chapter 3. Yes? Okay. And I think I asked someone to write down, we talked about the different types of compound microscopes, true, false?
Yeah.
True.
Examples of different compounds.
And where we use them, right? And how we use them. Okay, so I'm gonna just restart with listing, right? We talked about a simple microscope. And we started to talk about compound microscopes. We talked about the total magnification and the resolution and how those are really important features of the compound microscope. Right? Total magnification, how do we determine that? Well, we multiply the two, what two things do we multiply together?
The ocular and objective lenses.
Correct, the ocular and the objective that we're using at that moment. And for you guys, as we talked about in lab, it's almost always going to be 1000x, right? Because you're almost always going to have to use that immersion oil lens, and the ocular lenses are 10. Older microscopes, you might get a 5x or a 15x. Really expensive microscopes, you might get a 20x eyepiece. So you do have to at least check whenever you're using a new microscope. And the resolving power, remember, is the distance, the minimum distance necessary so that we can see two objects as two separate objects. And that's almost just as important as the magnification, because if you can see something really large, but it's blurry and you can't tell what it is, then that's not really of much use to anybody, right? So in general, all the compound microscopes that I'm going to list, their maximum total magnification, if you have all the special lenses, is going to be about 2000 times the size of life. And the maximum resolution is going to be 0.2 micrometers. So 0.2 millionth of a meter, right? So a 2/10 of a millionth of a meter, very, very close together. Now, we in lab, of course, use the compound bright field microscope. And one of the advantages of that microscope is that you can fix and preserve the specimens to see them later, right? So that was one of the big inventions. Yeah, go if you want to pull those closed, go ahead. It's totally fine if you can't see. But that's also one of the disadvantages. The specimens must always be stained, right? It's very difficult to see live organisms with a compound bright field microscope. You might remember from Bio 1, if you looked at protists, you were constantly adjusting the contrast and keeping the light low. And if they weren't photosynthetic and didn't have a pigment in them, they were just gray and it was hard to see, right? The compound dark field microscope, I think we kind of were talking about that barely at the end of class last time. Dark field, because when you look in there, the background is completely black and all you see is reflected light off the specimen. So you don't see anything until you put your slide under there. One of the advantages of the compound dark field microscope is that you can use it to see live specimens. And you can also use it to see things that are unstained. Right? So that's an advantage, right? However, with the invention of the phase contrast microscope, compound microscope, I'm going to stop writing that because it's repetitive, right? I think we also talked about this. The light gets polarized in the condenser. So you're changing the interactions between the wavelengths of light, and you're limiting the refraction and the reflection. And most of the time with a phase contrast microscope, it's a dial on the condenser. And so you can adjust how much of the phase contrast that you want. This microscope is like a combination of these two. And if you switch out the light source, remember the last one I had on here was fluorescent microscopy. You can purchase one where you can change out the bottom component and take out the white light bulb and put in a UV light bulb. And now you have like all 4 microscopes in one microscope. Right? Awesome. Except it's going to cost you about 4 times the amount of that microscope, right? Just as a reference, the scopes that you guys are using list for about $2,500, $2,600. The ones we use in lab, a really nice phase contrast microscope, brand new, that has that light changing capacity, is usually in the range of $8,000 to $10,000, right? So it's a lot more expensive piece of equipment. And so you find them less frequently in clinical settings, more commonly in research settings because of the expense and the maintenance. But advantage of the phase contrast is you can use it to look at things that are stained. You can use it to look at things that are unstained. You can use it to look at live specimens. And the last sort of super advantage is you can see both the edge and you can also see internal features if the cell is large enough for you to be able to see those internal features. I'm going to go ahead and put D on here, put the fluorescent microscope on here, fluorescent microscopy, before we look at the pictures again. You're going to need to stain with fluorescent dyes. There's fewer choices. They're usually multi-step staining procedures, more than what we did the other day in lab. Obviously, the light source is going to be changed to UV light. It works best if you are in a dark room or someplace where you can be in complete darkness. Sometimes people don't have the capacity for a dark room, so they have like a blackout curtain that comes just comes around the microscope, so that just the operator and the microscope can fit in there, like in a regular lab. Advantage with this is you can get better specificity. You're not staining everything, you're only staining the thing that you want to see if it's there. And if it's not there, you won't see any of the colors. It'll just be a dark background. So that is useful for specimens that are in low number, or I'll say low quantity. Right, so I'm trying to find something that's rare or hard to find. And I'm trying to increase the chances that I'm going to be able to see it by just staining that one thing. Because if it's not there, then it won't be there. But if it is there, I'm going to catch it. All right, so let's look at some pictures. Let's see if I can do that. They are small here, but you can magnify them when you look at them in your textbook. These are pictures from your book. It's page 69 and page 70. I just like to use these because it just makes it easy for us to take a look at them. So here's a paramecium, which you've all seen before in a bio textbook or under the microscope. And this one happens to be stained. Right. So this is what it looks like with a compound bright field microscope. And you can see the magnification is 230 times the size of life. It's something to start paying attention to, especially in chapter 4, which we're going to start today, in that those magnifications give you some indication of what type of microscope was used and how small the thing you're looking at really is. Here's a paramecium with a dark field microscope. Can you see how we have so much more internal detail that's available to us? We can see the macronucleus, the micronucleus, we can see the gut, and we can even see some of the cilia. Now, whoops, let's try again here. This guy, let me move it this way. No, guess not. I want that one to be bigger. It won't let me make it bigger. All right, well, we'll just do this. And then I think I can magnify it. Can you still see it? No, nothing. It's so fragile. I don't know why I can't make that bigger, but let's try this feature and put it here. There we go. I'm trying to move it over. Won't let me. Okay. So this is actually, I was wrong. The image that I showed you was not dark field, but it was the phase contrast, which is the better image anyway. So that's fine. I thought they also had a picture of fluorescence here on the bottom of this. Can you see this over here? Right. There's an example of a fluorescent, using a fluorescent microscope. Can you see that they've used 3 different colors of dye? We've used a blue, a green, and a red. So we can get, those are eukaryotic cells. You can see the nuclei are red. So those are really large cells. And we're only magnifying 400 times the size of life. But you can get more, like I said, you can label specific items, or you can find things that are rare to find. All right, let me get out of this because it's annoying. I think we should also mention confocal microscopy. This one up here in the corner. It's a version of compound microscopy. So what was I up to? E, right? E. Compound break field phase, fluorescent. B, C, D, E, yeah. Again, this is mostly used in a research setting, not so much in a clinical setting. And the specimens are going to be stained with fluorescent dyes again. However, instead of using UV light, you're going to illuminate those dyes with lasers, and you can use different colors to excite different colors of the dyes. And that's going to be fed through some sort of a computer. The microscope is going to be attached to the computer, and you're going to actually be able to manipulate the image a little bit more to expand certain areas of the image. And you can also take video off of the image. So this is primarily used to visualize cellular physiology. What I mean by that, so some of you've had A&P, yes, no? Yes, okay, so you've learned about a muscle contraction, maybe, or how a nerve impulse travels along a neuron, and then when it gets to the end, it gets converted from an electrical stimulus to a chemical stimulus. That's how we know this, right? People have taken time-lapsed photos a long time ago before they could do it with a live stream video, and they have labeled the calcium, and we can see the calcium hitting the sarcolemma, which is labeled a different color, and we can watch it actually happen, right? So a lot of those processes that you think, how could we know that the calcium release, you know, the acetylcholine causes the calcium release, which causes the myosin heavy chain, you know, to put its claw on there, and the ATP to pull that, that's how we know, right? So that's used a lot in any time it's a process or a step-by-step thing that someone's trying to study. Also think like mitosis, meiosis, cell division of any kind. All right, this next box, this box is electron microscopes. So that's like a whole other level of microscopy. And they generally come in two formats, okay? One that we call the transmission electron microscope and one that we call scanning electron microscope. And then there are modifications of those that do very specific things. They're called electron microscopes because they utilize the particle form of light rather than the wave form, which we've mostly talked about before, the wavelengths of light that are illuminating the specimen. This has to do with something that can generate enough of electrons that can actually poke at the specimen, right? So they're going to literally bounce off of the specimen, and that action is going to be recorded and turned into an image. This is a machine that takes up a small room, takes up a closet, okay? Usually you have to be a specialized operator in order to use the electron microscope because it's complicated. There's a lot of computer parts that go with it. And the staining procedure is much more intensive. That's going to take you 3 to 5 days of lab time to actually stain the specimen. And then you need someone who, in the case of a transmission electron microscope, that can slice it ultra thin on a machine called a microtome. All right. So I'm going to put particle, let me spell that, particle form of light. So we think about the protons and the electrons. The staining is going to be with heavy metals predominantly. Silver, gold, things that will withstand the bombardment with the electrons. The staining procedure usually takes a few days. If you are going to use a transmission, I'm just going to put EM for transmission electron microscope. Those will be just abbreviated as TEM, right? Later. The specimen has to be sliced, and so you are viewing the specimen in cross section or longitudinally. Now, of course, we have the capabilities of having a computer that can reassemble that for us, and then you can rotate it and look at it in 3 dimensions. This is a picture of a virus. All viruses have to be, if you want to see a virus, viruses, cell organelles, cell membrane. Any of those things, if you want to study those, you need an electron microscope. That's a picture of a coronavirus, and you can see it's been sliced, so you can look inside of it. You can see the spike proteins on the outside and the nuclear material on the inside. It looks like it's attaching to a eukaryotic cell there. And someone that might have been photographed because someone was trying to study how the virus attaches and which receptors the virus attaches to on eukaryotic cells. The other thing I will say is about a transmission electron microscope, the maximum magnification, the total magnification is 1,000,000 times the size of light. That's a lot more than 2,000 times the size of light, right? So I'll just write it over here. Maximum magnification is 1,000,000, 1 times 10 to the 6th x. And the resolution, I believe, is half a nanometer, 0.5 nanometers, I think. Which again, remember, a millimeter is 1,000th of a meter, a micrometer is 1,000,000th of a meter, a nanometer is 10 to the -9 of a meter. Okay. The main disadvantage of using a transmission EM is all the prep that you have to prepare it, and then you have to kind of turn it over to someone who can slice it for you, and then you usually sit with the electron microscopist while they are looking at your specimens. It's not the same kind of microscope that we have where you just look in and turn the dial. As you might imagine, there's a screen, and like I said, it's usually attached to a computer, and so then you can capture the images in olden times, we had a physical camera attached to them, and we had to take rolls of slide film and then develop the negatives, and then decide which ones of them showed the best of what we were trying to measure or identify that we would want to put in a research paper or on our poster. And then we would have to take them somewhere and say, can you make these 8x10 glossy photographs, right? Obviously a lot easier now, but no less expensive, right? The other type of major type of electron microscope is one we call a scanning electron microscope. And now they magnify almost as much. We used to say 100,000 times the size of life, but I think there are scanning that can now do a million times the size of life. 6x. But with the scanning, it's like it sounds, we're looking at the surface of the specimen. We're not looking inside like we did with the transmission, because we're literally slicing it open and looking inside. So every picture that you've seen of a taste bud, for example, or maybe a hair follicle, a lot of those are scanning electron micrographs because they're showing you how all the pieces fit together on the surface. This is an algae, this is a diatom that you might have looked at under the microscope back in Bio 1, right? I don't think we had this particular diatom, but I think you can see really easily these calcium or silicone discs, I guess they're calcium in this one, that are laying around the top of the organism, which is soft bodied and inside of that you can't see, right? When you looked at it under the microscope compound, it might have just been a circle, right? Like Cosmerium, right? It was just like two circles together. That's really what it looks like. Right? The resolution is less here. I think it's 2 micrometers or sorry, nanometers. I think it's 2 nanometers, but I'm not sure. Again, why bother telling you about all those? We're not going to use all of those. We're only going to use the compound bright field microscope, but many of the images that you're going to see in your textbook are these other types of microscopes. So it's, you know, it's kind of to your advantage to at least know what they are and what they're used for. What do you have to know for the test? Well, for the test, you have to know the applications, like when you would use which one, right? Or if the question said, which of the following microscopes are used to look at viruses, you have to be able to tell me that it's at least an electron microscope, right? No, this is the study guide. All the notes that you're taking and all the notes, there's a few, I think for chapter 4, there's like a note outline for you to kind of fill in as we go along. But everything that we've gone over is fair game for the exam. So I would focus on the notes, not so much on the connect questions. Those are to help you with the practice and the memorizing, because there's a lot of memorizing, right? Biology just has a lot of memorizing things before it ever gets interesting, right? So that is specifically, like I'm telling you right now, asterisk, star, red pen, highlighter. For this section, what you need to know for the test is you should be able to tell me the different types of microscopes, the dyes you might use, anything that's unusual about them, right? How you might stain a specimen, what the magnifications are, and what you would use them for. So like everything that we just went over. Does anybody have any questions about any of these? Off the top of your head right now, interest or otherwise? Yes. All right then. I'm going to erase and we're going to move on. All right, so the next section. of the chapter, sort of the last section of the chapter, is about making specimens and specimen preparation, which you already know a little bit about because you've done this in lab, right? But it's probably a good idea for us to go over it in very specific detail here as well. Just looking to see if I have a. Oh, well. Okay, so this section is going to be specimen prep and staining. And this is, like I said, this is kind of the last section of chapter 3. So the specimen prep you already know a little bit about because, like I said, we've already done it. The first step of that is to make a smear. Right, very highly scientific word there, right? And again, why am I going over this now? Because these questions are going to show up on your exam, even though this is a lab activity, right? Because we don't have lab practicals in this course. So, and it's right here in your textbook, and we're just going to go over how we do it in lab, and it's sort of to reinforce what you're doing in both places. So to make a smear, remember, it depends on if your source is in a broth or if it's on a solid, which we refer to as agar, right? So the broth is the liquid, obviously. The agar is the solid. So remember, we used our aseptic technique with our Bunsen burners so that we would not contaminate the specimen, right? And if we're taking it from a broth, that's gonna be a little different than if we're taking it from the slant. So when we take it from the broth, it's just, we're gonna dip in there, right? And we're going to put it on our slide. I'll even draw the wax circle for you, and we're just gonna smush it around. Ideally, we would like that broth to be slightly cloudy when we put it on our slide, which is why I asked you guys to take 3 to 5 loopfuls to get it to be enough on your slide so that you could see it, right? Based on your experience from Tuesday, when I hand you the next broth, you may think, well, I'm gonna put 5 to 10 loopfuls on because I didn't see diddly squat, right? So that's kind of up to you as the end user of the broth. Now, the bacteria I gave you, even though it had been growing from last week, that Staphylococcus epidermidis, it's not super fast grower, right? If I give you E. coli today, that broth you're going to see immediately is almost beige in color because it is so thick in there. So it sort of depends on how cloudy the tube is that I hand you, okay? But when you do a broth, you don't need the water. Obviously, when you take from a solid, remember, we're going to go in there and touch, not scrape. When we go from a solid to make our smear, we're going to need to add some drops of water first so that we can smear our bacteria around in the water. Then we dried the smear. You might, of course, you remember we used the like the hot plates basically that are over there on the side of the room. And once it was dry, then we fixed the smear. And in here, we're going to use heat or the flame to fix. We are not going to use alcohol. And again, I'm just drawing this for the sake of it. All right, so we're going to put our slide in there at about a 45-degree angle. And the fixing of the smear does 2 things. So this will help you answer that question in your lab report. The fixing of the smear does 2 things. First thing, it kills, well, let's say the first thing, the most important thing, it sticks the bacteria to the glass of the slide. And this is 2 parts. It is an ionic attraction. Right? Bacteria are negatively charged. The glass is slightly positively charged. So they sort of generally kind of stick together. We're enhancing that. And the other part of this is we denature the proteins in the bacteria a little bit, some of them, which proteins are sticky. You've made eggs before. Yes, you've cracked open an egg and it's stringy. That's because it's mostly protein, except for the yolk. The white is all protein and it's sticky. And so we activate that stickiness by heating it up, and that's going to help make it stick to the glass. The second important thing that fixing does is it kills the bacteria. And that's important because we gently dried them so that they wouldn't get so distorted, but now we need them to be dead because we don't want to transmit them around the lab or contaminate something else. So we need them to die officially. So those are the two major functions of fixing the smear. I'm sure you remember I told you, maybe not, because it was sort of toward the end. If you forget to fix, which you didn't on Tuesday because I was reminding you constantly, but you might forget today, I'll probably shout it out a few times in lab, but certainly by next Tuesday, right? It might slip your mind. If you don't fix when you put the stain on, the bacteria is going to stick more to the stain than to the glass. And so when you wash with water, which is also polar covalent, it's going to take those bacteria and the stain and put them all in the staining pan. And you will have little to nothing to look at on your slide. Okay. So fixing is, I can't emphasize enough how important the fixing step is. All right, so we make the smear, we dry the smear, we fix the smear. Now we can apply the stain, number 4. And depending on what the charge of the stain is and how many different stains we put on, those staining procedures go into categories. The first is what we call a simple stain. And simple, like simple microscope, means one dye. Okay. There are 2 versions of the simple stain. We call them a positive stain or a basic stain. Let's do positive first. Also known as a basic stain and a negative stain. Also known as an acidic stain or an acidic dye. I know that's complicated because basic and simple in everyday language mean the same thing, right? But they're not interchangeable in here, right? You're doing a simple stain. Technically, you were doing a positive simple stain on Tuesday, right? Reason being, we were using a dye. This single dye, the color ion in the dye is positively charged. Meaning, if I draw bacteria here, a couple bacteria here, the bacteria cell wall is slightly negatively charged, because remember, bacteria have an outer coating outside their cell membrane, but cell membranes are also usually negatively charged. And so when I applied the methylene blue that was positively charged, right, it's going to stick to the outside of the cell and make it look blue. On my diagram here, I'm just drawing it on the outside, but remember, bacteria are 3-dimensional, so really the whole thing is covered in blue, right? I know the bacillus looked better than the staph for us the other day. But conversely, if my simple stain is a negative stain or an acidic dye, that means the dye is negatively charged. And if I put a negatively charged dye on top of a negatively charged bacteria, I really wish I had my black marker, but I don't. what that's going to do is, because the dye is negatively charged, there's going to be a tiny little space between where the dye is sitting and the bacteria, right? I'm exaggerating it on the board. It's like obviously a microscopic space, because when you looked at the negative staining slides that I had on the side of the room, right? We say the background is stained and the cell is clear. Again, that would also look better on the bacillus than the staph that we had. With a positive stain, a positive simple stain, or a basic simple stain, the cells are stained and the background is clear. Now, if you put a lot of bacteria on there and it's so chocker full of bacteria, you can't see much of the background, the white background behind it, right? But if you drive out to the edge where it's not so crowded, you guys can see the cells individually with some white space behind them. This is the easiest type, quick and dirty. It basically just tells you if there are bacteria there, right? You can tell their size. I could try to make you measure them. You can tell their shape, which we refer to as their morphology. And you can tell their arrangement, which this is as good a time as any to talk about these things, because I'm going to try to be grading your labs today. And I'm going to be looking for you to use the correct form of these words, okay? So you might remember that the first slide we looked at on the first day, I'm going to write this, I'll put it over here. Let's put this on top. So you might remember that the first day when we looked at a slide, how many different shapes did I tell you you were going to see? 3, right? So shape equals morphology. I know, you're just like, you just wrote that. Yeah, I'm trying to get you to remember it, right? So I'm repeating it a lot. Shape equals morphology, and you have 3 categories of shape to choose from. That's it. We're not going to see any square bacteria in here. Go to graduate school, learn all about it, right? You are going to see what we refer to as coccus. Those are sphere shaped or round. I need you to use your big boy, big girl science words and write coccus, not round, on that line in your lab book, right? I've already waved my magic inoculating loop over you in lab when you weren't looking, so that you would use your science words, right? If you don't use your science words, I'm going to mark it wrong on all the labs, and you've got to learn this distinction, right? It's hard at first, but it gets easier. So we have that shape. We also have the bacillus shape, okay? And that one is rod shaped. They can be short, they can be long, but it's like a hot dog. And the third shape category, we actually call curved. And there are subcategories of curved that go with some Latin words. There are vibrios. Those are comma shaped. I don't want that. There are spirillum, which is the one you saw last time, or 2 times ago, a week ago, right? Multiple curves, let's just say, multiple bends in the bacteria, right? And the third category of curve is something called a spirochete. And these are actually spirals in 3 dimensions. They're round like this. Syphilis is a spirochete. Borrelia that causes Lyme disease is a spirochete. There's a leptospira that causes urinary tract infections, particularly in animals, or unless you're camping and drink unfiltered water. And they are actually spirals. Under the microscope, they'll look like this though, right? Because we're squishing those spirals flat. They'll look more like a zigzag. Again, you're not going to see any of these commonly. Most of these are pathogens. And so we're not going to let you touch them or stain them or make smears out of them. You're going to see coccus and bacillus over and over and over again. This is where the tricky part comes, okay? That's line number 1. So under the picture, when you are in your lab book for a simple stain, right, under the picture, it's going to say in capitals, Staphylococcus epidermidis. And because it's epidermidis, because it's a printed thing, they can write it in italics, right? I can't, so I'm going to write it like this with underlining 2 words. Remember, we talked about in lecture last time, that's how we name things, genus, species. And you've made some sort of a drawing in here, right? I don't know, something like this, maybe. Beautiful. And then under here, there's a line that says X for total magnification. All right, that's going to be 1000. And then under that, it's going to say morphology. And then it's also going to say arrangement. And this is one of the difficult things, because in the first lab or 2, I think on negative staining also, they leave them as separate lines. But today's lab, we're going to do the gram stain lab. You're only going to get 1 line, and it's going to say morphology and arrangement. It's just how we try to make you learn it. Now, morphology is just the morphology. So what shape are these bacteria? Coccus. So I'm going to write that here. Right? The arrangement is how are they next to other Staphylococcus, other coccus in this image that I've drawn? Are they all by themselves? We would call them single. Are they always in pairs? Then we're going to add Diplococcus to the word. Are they always in sets of 4? We would always. Are all of them that we see, or most of them, in sets of 4? Then we would call them a tetracocci. Or are they always in a chain? Or are they always in a clump? And those are pretty much your choices. The coccus has the most choices, so don't panic. Okay? Yes. So single. And you use single if you see almost all of them are separated from each other, and single. You use the word Diplococcus. You put this on as a prefix, right? So these are arrangements of coccus or cocci, because that's the plural, right? So they can be single. They can be always in pairs. Sorry, I was drawing a rod. And there are some bacteria that are always in pairs like that. And so think about the 5 eyes that we talked about at the beginning of chapter 3, right? So we inoculate, we incubate, we isolate, and then we inspect. And this is part of that inspection, that microscopic inspection. And if we see if you're a clinician, if you're a clinical microbiologist and you isolate a Diplococcus, that puts that big, what is this bacteria mystery, that puts it down to about 6 things like that, the day you look at it under the microscope, right? So it's a big deal that I can know the difference. They might be in chains. If they're in chains, like this, in a line, We call them strepto. We put the prefix streptococci. And if they are in clumps, we put the, like the ones that you saw, we call them staphylococci. And I get it, this is confusing. Sorry, it's too bad. We're gonna practice a lot, and I get it, it's confusing. When I'm using the arrangement word, I make the prefix is lowercase. And I repeat the shape word, the morphology word. And you're like, but you just said, I know, it's just how we do it, right? So this case would be, they were in clumps most of the time. So I'm going to put little s staphylococci because I saw more than one of them. It's the same word as its genus name, right? But when I'm talking about this particular bacteria, I use the capital when I'm saying its genus name. And the best like analogy I can do is this, right? There is a girl, there's a woman in my Bio 22101 that wears glasses. Right? That's a general term. I've got 1, 2, 3, 4, 5 of you that are wearing glasses, right? That's like saying coccus, right? A woman that wears glasses. 6, you're gonna keep them on, right? That's the generic one. But if I say Rashida Bell is in my 22101 section, that's her in the back in the green sweatshirt. She gets a capital S, right? That's just because I already know her name from before. That's the best analogy I can give you. When I'm talking about just who's on the slide, I'm describing them generally, I use a lowercase. When I'm naming them, I'm saying this bacteria is Staphylococcus epidermidis, capital, lowercase, underline, underline. Right? Your lab book is always going to tell you what you're looking at, right? And sometimes it's a big clue to either its morphology or its morphology and arrangement. You just have to spell it correctly and make it the first letter lowercase. It's important difference because this isn't the only bacteria that has this morphology. You might know the bacteria MRSA. You've heard of that before, right? In the hospital setting, or you've read about that before. And it's MRSA, all capitals. That stands for methicillin-resistant Staphylococcus aureus. That's a different Staphylococcus, and it will kill you, right? Or can kill you. So I have to spell its name correctly if I'm naming that one, but it is also a staphylococci. When I look at it under the microscope, it's not one of these that limits it to 6 bacteria. It's a staphylococci, which limits it to like maybe 20 different bacteria. And I have to do more tests before I can name it, right? I have to do those biochemical tests, those genetic tests, those immunological tests, that identification step of the 5 eyes before I can say, who is this, right? The same rules apply for bacillus. However, because of its shape, you will mostly see it as a single or as a diplobacillus or diplobacilli, or in a chain, strepto. Because it's rod shaped, it doesn't usually form a clump. Yours were clumpy because you took a lot of them, even though you were trying not to, right? It's not super important to me that you choose the correct one. It's just you have to choose something and put it on this line, right? Like, meaning, If they're staph, they're gonna look like that. But as far as the bacillus, if you put single, but most of them were in a chain, you can put single comma streptobacilli if you want. That's okay too. You have to put something on the line. And then, like I said, in today's lab, you're only gonna get one line, and it's gonna say morphology and arrangement. So then you only have to use one word. You don't have to repeat. You can just put little s staphylococci. These guys are always single because of their shape. They don't form clumps or chains or diplos because they're curvy. And so normally they aren't touching each other, right? When you isolate them. Okay. There are more stains though that we have to mention. A few. We're almost done. So we have a simple stain. We have something we call a differential stain. The clue is in the word. It allows you to tell the difference or distinguish between two different kinds of bacteria. And in this case, all the differential stains, they are based on types of cell wall. Again, this will make a little more sense after we talk about chapter 4, because we're going to talk about all the different kinds of cell walls. For right now, I'm just going to give you 2. Okay. One of them is that Gram stain that we mentioned before. The other day, I think I used the word Gram stain. Capital G, Gram stain. Okay. And that tells the difference between a Gram positive cell wall and a Gram negative cell wall. And that's important for diagnostic purposes as well as identification purposes. And the other major differential stain is one we call the AF for acid fast. And that too distinguishes between 2 types of cell wall, acid fast, we say, and non-acid fast. And they're like, okay, why don't we say plus and minus on those? Because we don't want, when we're verbally communicating that over the phone to a physician, we don't want to confuse the words. And if we say positive and negative, we always say positive and negative, it could easily be confused that they thought we said the Gram reaction. And this is very diagnostically different because the acid fast bacteria that cause diseases are tuberculosis and leprosy. And those are treated very differently than a bacteria that's just E. coli. Okay? So we use different language so that that won't be confused when we're writing it or when we're speaking it. It won't be misinterpreted. So we're going to be Gram positive, Gram negative, or acid fast or non-acid fast. And those are also different types of cell walls. Then there's a group of stains we call the special stains. They get special because they're for special things that bacteria might have. Third group are special stains. And these are usually for special structures. And that means things that most bacteria don't have. Capsules, flagella, spores, or endospores. The capsules and flagellas are simple stains, meaning one dye, right? But they might require some special procedures other than just fixing, right? Or the staining procedure might be slightly different, maybe longer, maybe you have to use heat, maybe you put it in the refrigerator. The capsule and flagella, that's why we call them special, but they're single dye stains or simple stains. The spore stain is a differential stain because it allows you to tell the difference between the spores and the bacteria. You guys all saw spores on Tuesday, and they were kind of whitish, but also kind of bluish because we didn't do a spore stain. We just did a simple stain. Now, I would like to show you some pictures of the different stains, and then we're gonna call it a day for the lecture portion, and we're gonna go to lab. So let's take a look here. Wait, I want to screen share now. Let me share, please. Okay, so this is in the PowerPoints, which are, oh, sorry. This is in the PowerPoints, which are already up there posted for chapter 3. They're all the pictures, right? Like I didn't draw a picture of a microscope or a compound electron microscope. So some positive staining, negative staining, and some of the dyes that we might use for those things, right? You saw we used methylene blue. We can easily use the first dye of the differential stain, which is a crystal violet. It depends on how you make it. If you make it acidic or if you make it basic, you can use it as a positive simple stain or a negative simple stain, right? And that's similar to what we saw under the microscope. This is a little more purpley than we had, but nonetheless. And here are some pictures of some of those other stains. In the center, the differential ones are the ones I want you to mostly focus on. Can you see pink and purple here? Right, so that's a gram stain because we use 2 different dyes, a red one and a purple one. This is an acid-fast stain. All these little pink blobs are the acid-fast bacteria. And then this is a spore stain that's actually stained for spores. And you might be able to see how the spores are all red? And then the whole cells are kind of purpley pink. Sometimes that'll be a green and red differential of dye. And then lastly, we have, you can use India ink to do capsules. Can you see in that far column how there's a little tiny rod inside of a clear space and then the background is stained? That's because the capsule resists the dye. Sometimes that's done as a differential stain where you stain the bacteria first and then you stain the background. And then the flagellar stain on the bottom, that's Proteus vulgaris. Can you see all those little spidery things, like spidery things coming out? We don't do that stain in class because it takes too long. It takes like a whole day or hours because you have to keep applying the stain, and it's hard to do. And most of you won't get a good result. And even the slides I give you that somebody else prepared, most of us aren't going to see the flagella. So, right. Some of these we're going to do. Gram stain, acid fast. The rest we're just going to look at on prepared slides. Okay. Make this stop screen sharing. Oh, I think I forgot to share that with the people at home. Maybe. Let me just put that up there for a minute and then stop. All right, I think we're gonna stop here, even though I would like to start chapter 4, but I think I'll just end here. Do you want to stop? Yes.